Picture this. In a quiet hospital room, doctors used a single CRISPR-edited cell to alter the course of an infant's life. How close are we to being able to rewrite the future for all rare diseases?
Today's date is 2025-05-16. Let's dive right in. The case we're talking about is actually the first time a custom CRISPR gene editing therapy was used to save a newborn. This happened at the Children's Hospital of Philadelphia, didn't it?
Yes. The newborn, KJ Muldoon, was diagnosed almost right after birth with CPS1 deficiency, a rare genetic disorder. Without treatment, toxic ammonia builds up and it can be fatal. Up until now, the only real option has been a liver transplant. But in this case, doctors used rapid DNA sequencing to pinpoint the mutation and then moved to correct it at the genetic level.
And this wasn't a standard CRISPR treatment, was it? They actually created something specific for KJ's mutation.
That's right. The team, Dr. Rebecca Aarons-Nicholas and Dr. Kiran Musunuru among them, developed a custom CRISPR base editing therapy. Everything from diagnosis to treatment took just 6.5 months, including design, testing, and even getting FDA approval. The therapy was delivered directly to KJ's liver cells to address the root cause.
The speed is striking. Normally, drug development takes years. Do you think this kind of rapid development is realistic for other conditions? Or was this a special case because of the urgency?
That's a crucial point. Here, the urgency and the clear genetic target made it possible. If we tried to scale this for more common conditions, we'd face much bigger challenges, like genetic complexity, the need for ultra-rapid diagnosis, and not to mention regulation. Right now, the infrastructure for such personalized treatments isn't widely available.
So given how complicated and costly these therapies are, does that mean they'll be limited to wealthier countries for the foreseeable future?
Probably, at least for now. The expertise, facilities, and funding needed are significant hurdles. To make these therapies accessible worldwide, we'd need international partnerships and dedicated funding to build capacity, especially in lower-income regions. Otherwise, access is going to be very uneven.
And for the families who do get access, what comes next? Gene editing sounds definitive, but are there risks that might show up years later? What kind of long-term follow-up is needed?
Ongoing monitoring is critical. Since these therapies are so new, we don't have the full picture yet. There's a chance of off-target genetic edits, immune reactions, or unexpected complications. Patient registries and regular checkups are going to be essential to spot and manage any issues as they emerge.
So we have a newborn saved by science, but the future is still full of questions. How quickly can we scale this? Who will benefit? And how do we make sure it stays safe?
Exactly. This marks a turning point. We're moving from just managing symptoms to actually fixing diseases at their genetic roots. That shift is going to challenge healthcare systems, regulators, and whole societies to rethink how they approach rare and perhaps even common diseases.
It's a big moment for medicine, but it also opens up deeper debates about cost, access, ethics, and what it really means to change someone's DNA.
Thanks for joining us. Today, we saw how a single genetic edit can change a life and raise new questions about the future of healthcare.
Stay curious, keep asking questions, and we'll keep bringing you the stories that matter.